Hello everyone, welcome back to our discussion on mutation. So now we are going to discuss about the first type of mutation, which is a chromosomal mutation. And in chromosomal mutation, we'll be discussing about the changes, or what are the changes, or what are the consequences of having change in chromosome number, and what are the consequences of having change in chromosome structure. So, chromosomal mutations or chromosomal aberrations, as we said, it will be divided, or it will be discussed in uh, two different. It will be discussed in two different heads. One is the change in chromosome structure. In that, we'll discuss about deletion, duplication, inversion, and translocation. And in change in chromosome number, we'll be discussing about aberrant euploidy. And in aberrant euploidy, we'll discuss what is monoploidy, diploidy, polyploidy, and aneuploidy, where you have trisomy, monosomy, and nullisomy. So these are the difference, or these are the chromosomal mutations that we are going to discuss in this particular session, coming under change in chromosome structure and change in chromosome number. So first we'll take the first type, which is change in chromosome structure, and in that we have four different types: deletion, duplication, inversion, and translocation. So moving on to the details of one by one. So this is just a representation of what are the things that we are going to discuss. So this is a chromosome, and this is a chromosome. They haven't shown centromere, etc. But consider there will be a centromere here. This is a sister chromatid. But this is a sister chromatid, a particular section or a particular chromosome which is shown here, and this is a wild type, means a normal one. So here we are going to discuss the different types of change in chromosome structure depending upon this colored bands. So we'll be discussing, or we'll be looking this particular different types depending upon the change in this color bands. So first type is, you can see this entire diagram is divided into two shades. This is the light, this cream color shade is the thing which is called as loss of genetic material. That means due to this type of chromosomal mutation or change in chromosome structure, the gene content will be lost from the chromosome. So that particular portion will be lost. In that you have deletion, and this missing chromosomes, that is the one which we'll be discussing in the second heading, which is called as change in chromosome number. So in this particular one, in deletion, what you can see is that, so this is loss of genetic material. That in this particular session, we have only deletion in this case, and you can see relocation of the genetic material, and that is duplication, inversion, and translocation. In all that, what happens is that there is no loss of genetic material. The genetic material is either shifted to another location, which is translocation, or it is shifted to another chromosome, which is translocation, or it shifts its position, which is called as inversion, or it becomes duplicated. So there is no loss in the three different types of chromosome change in chromosome structure, like duplication, inversion, and translocation. In deletion, you have a loss of genetic material. So we'll discuss one by one. So what happens here? You can see four colored bands here: pink, green, brown, and blue. So the first is deletion. In this deletion, you can see that this brown portion is deleted. As a result, in the deleted chromosome, when you compare it with the wild type sequence, this particular portion, so this might be having many genes in this. So all the genes which is present in this particular portion is now deleted. It is lost from the chromosome. And now coming to duplication. So in duplication, what happens is that you can see that this brown portion, or this brown segment, is duplicated, or this particular portion is duplicated, and it is now present in twice the content as that of the wild type. So in wild type, it was of this particular length, and then when this is duplicated, this portion will be represented twice. So that is what is called as duplication of a chromosomal portion, and that is a change in structure, and that is called as duplication. And the next one is inversion. Inversion, you can see that here also there is no loss of genetic material, but the order of the genes will be reversed. So here you can see in this, the order is pink, green, brown, and blue. But here, the center portion is just being this particular segments, they are inverted. They break and they shift their location in that. They shift their position in that. They have an inverted position in the mutated chromosome. Coming to the last one, which is translocation. In translocation, you can see that. So this is a piece of a DNA which is not belonging to this wild type. So this particular piece, it is from a non-homologous chromosome. A particular piece of chromosome from a non-homologous chromosome has shifted its location to this particular chromosome, and this is what is called as translocation or change in position. So these four: deletion, translocation, inversion, and duplication. These four comes under what is called as change in chromosome structure. So we are now discussing chromosomal mutation or chromosomal aberration. In that we have two sub portions. One is change in or chromosome mutation due to change in chromosome structure. Of that you have deletion, translocation, inversion, and duplication. And the chromosomal mutation due to change in chromosome number is either due to missing, can be due to missing chromosome, or due to extra chromosome. So this two comes under the second type, which is called as change in chromosome number. So we in this particular session we'll be discussing these four: deletion, translocation, inversion, and duplication. So now we'll move on to the details of each one. First one will be deletion. So deletion, in change in chromosome structure, the first one is deletion. So we are discussing this one where this portion is deleted in the new or the mutant chromosome. So a deletion is simply the loss of a part of one of the chromosomal arm. The process of deletion requires two chromosomal breaks to cut the intervening segment. So two cuts will be there. As a result, this particular portion will be removed. The deleted fragment usually do not have the centromere. It will not, because a small piece is being lost from the chromosome, and usually in deletion like this, that particular portion will not have the centromere. As a result, that particular piece will be lost from the chromosome. And the effect of deletion depends on the size. As I said, if there is only one gene, the effect will be less. If there are many genes, the effect of mutation will be more. A small deletion within a gene is called as intragenic deletion, which inactivates that particular gene. That means if this particular portion is a piece, you have a very big gene which is coding for a protein, and this particular the deleted portion is a small piece or a small part of that particular gene, that is called as an intragenic deletion. And in this case, it will be activating only that gene. That gene will be affected. And you have multigenic deletions where several genes. So if this particular piece contains many genes, that is called as multigenic deletions, where several genes are missing, and the consequence of deletions of these deletions are more severe than intragenic deletions. So deletions are usually identified by examining meiotic chromosomes under meiotic chromosome under microscope. And what can you see is that this deleted, if consider this particular chromosome is now at the G1 phase. During G1 phase, the deletion occurred. And now this particular segment, what happens is this particular DNA, consider that this is the, so this was a chromosome, okay? And after deletion, a portion of this was deleted. Okay, as a result, it becomes more smaller than the wild type because a portion is deleted. A portion has been deleted in this, so it becomes smaller. And this is if you consider humans, uh, we are having this chromosome, or we are having homologous pairs. And consider that one of the homologous pair. So if this is chromosome number 1, you have two chromosome number 1, and deletion happened in one of the homologous pair, and the other homologous pair is normal, the larger one, it is a normal one, and this is the one which is having that deleted portion. One portion of that is being deleted. So what happens is, this particular chromosome or these particular chromosomes, they enter, if it is G1 phase, they move to S1, the deleted one will replicate, and the other one, as every chromosome inside the cell will replicate, it will have such a structure, and they are moving on to the, or if these are the gametic cells, consider this deletion has happened in the gametic cells or germ cells, this particular chromosome, they will move to meiosis. And during meiosis, as we said, during prophase, because these are homologous chromosome, they will come close together, and they will, because they are homologous, they start to cross over. So when they try to cross over, what happens is that usually if these two were trying to cross over, they are having all genes in common. Okay, there is no dissimilar or dissimilarity if this was the case. But what happened in this was, this particular chromosome, now a portion of this particular chromosome is deleted. As a result, this is having a piece which is a portion of the chromosome is missing from this chromosome. So when they pair up, or when they start to cross over, such a structure can be seen, and this is called as a deletion loop. So here, uh, what you have to see is that this particular line is one sister chromatid, this is another sister chromatid. Okay, these are homologous chromosomes, and this is one sister chromatid, and this is the other sister chromatid. So this dark brown is one sister chromatid, and this two line indicates the two sister chromatids. These are homologous chromosomes, and this one is one sister chromatid and the other sister chromatid of this particular chromosome. So what happens is that this brown color, which you can see in the which is seen below, and you can see here these synaptonemal complexes formed, etc. So they are going to cross over, synaptonemal complexes formed. So this particular one, which is this particular sister chromatids, or this particular chromosome of the homologous pair, is the deleted one. So this is having something which is missing, and this particular chromosome is a normal one. For say, you can see that A, B is there, C and D is missing here. So this, in this brown, C, D is missing, and E, F is there. So this particular portion is missing. As a result, they do not have complementary regions here. As a result, they will not pair up. That is, the genes, the homologous portions of these particular homologous pair, the C and D are missing. As a result, because this is not present here, this C and D in the normal chromosome, not in the deleted one it is missing. In the normal or the undeleted chromosome, this particular portion, it will loop out, because it cannot involve in crossover, because in the deleted one, the homologous regions are not present. As a result, it will remain out from the crossing over. So to remain outside of the crossing over, or not involving in crossing over, it will loop, or it will form a loop, so that it is not involving in crossing over, because it is not having its homologous genes in the deleted portion. And this and the if you see such loops, you can say that this can be a deletion loop, and because the presence of this deletion loop will be an indication that there is some problem with the chromosome. So looping will not always indicate a deletion, because looping can also be seen in uh, duplication. So this is what is deletion. When there is duplication also, such a loop can be seen. So, if you are observing meiotic chromosome and or chromosomes, and if you observe a loop, definitely you can see that there is a change in the chromosome structure in one of the particular homologous pair. So it can be either deletion or duplication. So next is Now, so I think deletion and deletion loop, etc., is clear. And now we are moving on to the second type of change in chromosome structure, which is duplication. So this is what is called as, so this is what we are going to discuss now, which is called as duplication. So duplication is a process where in duplication, what happens is the process of chromosomal mutation sometimes produce an extra copy of a chromosomal region, and the duplicated regions can be located adjacent to each other. That means like this. This is called as tandem because this particular portion is duplicated and it is present here, adjacent to the duplicated segment, and that is called as tandem duplication. And if this extra portion is located, it should be located in the same chromosome. And if it is located elsewhere rather than nearby, if it is located elsewhere from the duplicated gene, that will be called as an insertional duplication. Okay, and a diploid cell containing a duplication will have three copies. So, three copies in the sense it is why they call say that there is a three copy. So, if I consider this as the duplicated one, so I am taking the homologous pair of this particular one, and I am marking it as A, B, C. Consider this B and C is duplicated again, and you can see B and C here. Again, you can see B and C here, and then I write it as this portion is E, and this is F. Okay, so this is the region which is being duplicated. B and C is duplicated. So this one is normal. A, B, C here. Uh, so I must D there. D, E, and F. So these are the chromosomes which are duplicated. This one is a duplicated one, and this one is the normal one. So what they say that you will have three copies of this particular chromosomal region. So that is true. If you take these two homologous chromosomes in a diploid organism, actually there will be only two copies. One copy in one homolog, this B and C in one homolog, and this B and C in another homolog. But when it is duplicated, you will have three of the segments, one, two, and 3. Two in the duplicated chromosome and one in the other chromosome. Okay, so that is why they say it has three. And in the meiotic process, the chromosome with tandem duplication. So if there is a tandem duplication, what you can see is that a loop consisting of the unpaired region can be seen. That's why I said, because when it is duplicated and when the other chromosome comes here. So if this is a particular case, what happens is this particular region is not having a homolog in this particular region. So this region will loop out when they try to form a synaptonymal complex or crossing over. This particular region will loop out, and that is what is called as a loop. And this is called as a duplication loop. The other one is called as deletion loop. The difference between a duplication loop and a deletion loop is that we said that in a deletion, the loop is seen in the normal counterpart. Okay, as you see here, the loop here is seen. The loop is formed in the normal chromosome, not with the deleted one. But when you see this particular duplication, the loop is formed in the duplicated segment. Okay, not in the normal one, because extra portion is in the duplicated one. As a result, the duplicated one will show, or the mutated one will show a loop in the case of duplication. So coming to change in the third one, which is inversion. So inversion, as I said, as you see here, inversion is this is what is called as inversion, that is just shuffling or flipping and reinsertion of chromosomal segment is called as inversion. And inversion is mainly two different types, paracentric and pericentric. So that is shown here. That is, if inversion does not involve, so this is a centromere region. The region, this is the centromere region of a chromosome in this particular case, and you can see that here there was an inversion. So this is a normal sequence A B C D E F, and you can see that in this particular inversion, this particular region has inverted. Okay, instead of DE, it is inverted. It has shifted. D has shifted here, E has shifted here, so that this particular inversion is not involving the centromere. Centromere is not involving, or centromere is not involved in the inversion. Okay, such type of inversions are called as paracentric inversion. And when centromere is involved, you can see that when these two gets inverted, that is when C is shifted here to this particular position, and or C and D, whatever it is, here you have to, and this particular is shifted towards this particular portion. So in this type of inversion, what happens is that the centromere is involved, or the inversions which are spanning the centromere also gets inverted in the case of pericentric inversion. If inversions are localized to any of the arm. In any arm, inversion can occur. And inversion, if it is localized to any of the arms, it is called as paracentric. And if it is involving the centromere, as a result, the inversion will involve both the arms of the chromosomes. And in such cases, it is called as pericentric inversion. And depending upon that, you have two types of inversion, depending upon whether it is restricted to the arms or whether it is spanning both the arms. And because inversions are balanced rearrangement, they do not change the overall amount of the genetic material, and usually they do not result in gene imbalance. There is no loss of genetic material, but other problems can arise, because when the genes are inverted, the problems arise when they try to cross over, or when the during meiosis, when they try to undergo crossing over and recombination. What happens is that the homologous genes, or the similar genes, will not be present. As a result, they will have to assume uh, uh, abnormal structures for crossing over to occur, and that attaining abnormal structures will result in abnormal chromosomes. And usually, this inverted chromosomes, they will end in most cases, you will not get, if crossing over or meiosis happens. Fertile gametes will not result from inverted chromosomes. So we'll take the examples of both the inversions here. So here first we'll discuss about the paracentric inversion, which means the inversions which do not involve the centromere region. So here you can see this is the normal one. Light yellow is the normal one, or light brown is the normal one, and the brown is the inverted one. Here this is not involving the centromere. This happens only in one arm. That is this B and D, it is getting inverted. As a result, the sequence becomes A, D, C, B, and E. Now, so this is the inverted portion. And as I said, inversion, as in inversion, there is no loss of genetic material, but when they try to cross over, so they try to cross over in such a manner that the homologous or the same locus comes in the same location, so they attain such structures. So you can see different structures. So this is one structure for attaining that. That means they will attain this same sequence as that of the normal one, and such structures will be obtained. And this structures will result in the formation of, you can see here, result in the formation of abnormal chromosomes like this, and this particular chromosomes. So this actually, they are homologous chromosomes. So this is the normal one, this is the inverted one. And in this case, even though both contain the same type of or same amount of genetic material, it is inverted. Actually, they are homologous, but because of inversion, you cannot call them as inverted. You cannot call them as now homologous chromosomes. They have to be considered as a heterozygote, and they are called as inversion heterozygotes. So this is a paracentric inversion heterozygote. They are called as heterozygotes because now actually they are having the same genes, but the order is different. So they are now not called as homologous chromosomes. They are said to be, said to have, or they are homologous but inverted ones, and they are given a name which is called as inversion heterozygotes. Okay, in that, the first case is paracentric inversion. So, what happens in paracentric inversion, as you see here, this light brown is the normal one, okay, and this one is the abnormal one, the brown one. So, what this brown or the inverted one does is that to attain this particular sequence or this particular to attain a position where the correct genes can be crossed. That means here, because of inversion, now B is, if they are pairing in this manner, now B has to pair with B, and that is not possible. So for pairing or crossing over to occur, B should align with B itself. For that to happen, they attain such a position where you can see that this particular chromosome is now coiling around. So when it coils, after A, you are having D. So now when this coils up here, D will come with D, C, and B, and then it goes for E. So the inverted chromosome is now attaining a different position here, and they will try to cross over. So here you can, if let me see, if I zoom in, you can see this particular region where crossing over has happened. So here you can see this particular sister chromatid has now crossed over with this particular one, and this particular one has crossed over with this particular portion. And as a result of this crossover, after this crossover is resolved, this particular chromosomes or this particular homologous pair, they will have new combinations, and we'll see how you read this new combinations. So this is the centromere region. So this particular sister chromatid, this is not involving in crossing over. So this has not involved in any crossover. Okay, the same is the case with the one which is inside this one, this which is moving inside, this is also not involved in any crossover. As a result, you can see that this will be represented as the normal sequence A, B, C, D, E, and this will be as the inverted sequence A, D, C, B, and E. So the outermost one of the light brown and the inner one of the inverted one has not involved in crossing over. As a result, you can see their sequence are the same. There's no difference. But the other one, if you take from here, the first gene is A, then it goes like this. You have B, and it is now crossover. So you should watch this carefully how I'm going to read this. A, then B. So this is now crossover with this. Now it has AB. After that, it has C here. Okay, sorry. It has C here, and now it moves to this particular point D, then E, and then again it moves where it will have A. Okay, so this is coming like this and moves downwards and A. As a result, this particular sister chromatid, because of this particular inversion, this particular sister chromatid is now having two centromeres, which is abnormal. Okay, it will have two centromeres at this particular after or during crossover. Okay, and now if you observe the other one, if you take the other one, you can see this is the other one from here. So this is also having the portion which is called as so this was the inner one, which is the other. So this was the outer one, and now the other segment is, if you read from here, you can see that it will have E, then D, then C, and now it is moving like this. It will have B, A, and E. Okay, and that is this portion, and that do not have, you see this is E. So I am going to read this particular portion. This is E, started the second one E, then I'm moving, you have D and C, which is light yellow. It is this particular portion, and it has crossover and moving down it has B. Okay, it does not have A here. Sorry, after B it is moving down like this, it is having E here. Okay, so this particular portion, if you read from here it goes from here moving down, it does not have a centromere, and this is what is called as an acentric fragment, and this acentric fragment will be lost. Okay, that particular portion, because it does not have a centromere, it will not be pulled by the spindle fibers. As a result, that is lost. Okay, and as I said, this is now as a tug, because this particular portion, as we read here, this is having two connections with the two centromere, and during segregation, the centromeres, the homologous portion, the centromere, they start to pull apart, but these are tied together. Tied in the sense, if you read it from here, this one, A, you can see here, I'm reading this particular portion, just watch A, then B, it moves down. You have dark brown in C, then dark brown in D, E, and then I'm moving down, which is having A with this centromere. So that is this particular portion which I read it now. And now what happens is during anaphase 1, the homologous chromosome, they try to separate. So this particular portion will be attached to a spindle fiber. This will be attached, and they start to pull them. As a result, there will be a break. This is called as a dicentric bridge, which breaks randomly. It can break at any point, here, any point. This is a random break which is shown here. Any point it can break off, and that will result in such structure. So this is also not equal. Actually, it should be having A, B, C, D here. This also should be having A, B, C, D here. But only two sister chromatids are normal now, two are abnormal. And that is what is shown after meiosis. Okay, you will have, after this crossing, after every type of divisions, you will have four chromatids like this. Of this, two are normal and two are deleted products. As a result, in any case, a fertile gamete will not be produced in this case. So the same as with this pericentric inversion. So in pericentric inversion, as we said, it involves or it is spanning the two arms, where the centromere is also involved. Here you can see this dark brown is a normal one, A, B, C, D, and you can see this C and B. This has now inverted, and this is called as the pericentric inversion heterozygous. And what happens during crossing over, as we said earlier, this will attain a looped structure so that they can pair up with the homologous pair. And you can see this looping is peculiar because the centromere is involved in this. Now, so here this one is not involved in crossover, and the last one, the inside one here, this is also not involved in crossing over, so that you will definitely have two products in normal, and the others after crossing over, because they are not having a chance to cross over. That's why only the adjacent ones usually cross over. As a result, the outermost one and the innermost one is not having, so this is the outermost sister chromatid, this is the innermost one, it is being looped, but they do not have a chance to cross over here. And after the two sister chromatids which are crossing over, you can see that if you take the second one, you can read it like this: A in dark brown, A, then B, now it has crossover here. So this is moving down, A, B, then it will be in light brown, D, and then to A. So that is this particular portion, that is A, B, C, and A. So this is like this: A, B, moving to this, which is A, B, C, and this is moving down like this: A, B, C, and A in light. Okay, the other one, if you take here, this is starting with D. Okay, and without involving C, it is again crossing over to this particular portion, and you have B, D, B, and moving down. Okay, after B, it is moving down here. It will have this will have C, and then last it will have D. So this is the crossover portion. That is, this is the one D, B, C, and D. Okay, so this is duplication of the D arm. This in this also you get only one normal product. In other two you have duplication and one will be an inverted product. So in any case, if inversion occurs, what happens is that uh, uh, in this case you can see that the four chromosomes are present. So gamete will be found, but it will have defects because chromosomes are missing. Only one is normal. In this case, a fertile gamete will not be formed because there is no pairing or a normal mitosis itself cannot happen in this particular case. Here, mitosis etc. can happen, that is meiosis 1 and meiosis 2 can happen, and gametes will be formed, but the viability of the offspring will be questioned because many of the genes are missing and some additional genes are present, etc. So, this is about inversion, and the last one is translocation. So translocation means in translocation, the two chromosomes trade their acentric fragments because in translocation the centromere will not be involved. A piece of one chromosome will be shifted to another homologous or another non-homologous chromosome. Acentric created by simultaneous breaks. Mainly two types of translocations are important or discussed. One is reciprocal translocation and the other one is Robertsonian translocation. We'll be discussing reciprocal in detail. A Robertsonian translocation is usually seen in rare types of Down syndromes, which is called as Robertsonian Down syndrome. And translocations are diagnosed by the apparent linkage of genes whose normal loci are on separate chromosome. So that is identified because some chromosome will have additional genes there which is not usually present there. So some protein might be produced in more amount than usual. Some proteins will be produced in less amount than normal. So that is how you identify whether a particular gene or a particular segment is translocated or not. And due to translocation also, you can have such a type of configuration during meiosis. So these are the normal N1 and N2 are the normal. They are non-homologous chromosomes, that's why you they are of different sizes and different colors. And one is a one if you take it as first chromosome, this one will be the second chromosome. And what happens is a piece from one is shifted to two and a piece from two is shifted to one. So these are the translocated one and these are the normal ones. So what happens is for during pairing, they will attain a cross-like configuration like this, so that the same genes or same loci they come together and a cross-like meiotic pairing can be seen. And after this pairing, they can segregate in two different manner. That is adjacent type of segregation and alternate segregation. Adjacent segregation means adjacent means nearby. T1 and T2 to one pole and N1 and T2 to one pole like this. That means they are getting segregated. These two will move to one pole and these two will move to one pole. And in such cases, you can see that there will be duplications, deletions, etc. But when it is alternate in the sense T1 and T2, like in the diagonal fashion, T1 and T2 is moving on to one cell and this to the other one. If it is segregation happens like this, if T1 and T2 comes, one will have a translocation genotype and this N1 and N2, as you see, they are normal. So this one will be translocated one and this one will be normal. And in this case, if alternate or alternate segregation happens, alternate in the sense these two are moving on to one cell, these two are moving on to the other cell. As a result, what happens is that this is complete, this is completely normal. This also will act as normal because of translocation, the genes are not missing, but only the positions have changed. As a result, both are complete and viable. In this case, because if this happens, some in some cases there will be duplication. The other cases there is deletion and deletion of some portion and duplication of some portion. As a result, there will be this will be in this case it will not be viable. But if this type of segregation occurs, the gametes will be viable. So with this, we come to the end of the discussion of the uh, chromosomal mutation due to change in chromosome structure. In the next session, we'll be discussing about chromosomal mutations due to change in chromosome number. Thanks for hearing.